Hello, I'm Caitlin O'Connell, and I'm talking today about some of my postdoctoral research at Rutgers University that so far has shown that episodes of fruit scarcity lead to muscle tissue wasting in wild Bornean orangutans. The rainforests of Southeast Asia are characterized as particularly challenging habitats for frugivores. These rainforests experience greater variation in annual fruit productivity compared to African and South American forests, as you can see here depicted in Van Schaik and Fannis figure from 2005. The island of Borneo is especially limiting, as we see lower overall fruit productivity there compared to Sumatra. This figure from Wick and colleagues shows Bornean study sites in blue and Sumatran study sites in red, and demonstrates that Sumatra consistently has a higher percentage of trees fruiting compared to Bornean sites. This low resource abundance has indeed shaped Borneo's fauna in various ways. For example, Bornean mammals have smaller body sizes compared to their counterparts on Sumatra, Java, and the Malay Peninsula. Given that orangutans are larger than most other mammals on Borneo, they have certainly had to find ways to cope with these really poor foraging conditions. They experience as much as a 70% reduction in energy intake in some populations when fruit is low. So they have a whole suite of adaptations to deal with low fruit. Behaviorally, orangutans are less social than most haplorhine primates and are less social compared to the other apes. Bornean orangutans are even less social than their Sumatran counterparts, which has been attributed to the lower fruit production on Borneo. During low fruit periods, orangutans also modify their activity budgets. They end up reducing the time they spent traveling, reducing the time that they are active during the day, and increasing the amount of time that they spend feeding. Morphologically, Bornean orangutans have more robust mandibles compared to Sumatran orangutans, which has been argued to facilitate the processing of much harder and more fibrous food compared to Sumatran populations. And finally, orangutans in general have the physiological adaptation of having an exceptionally low basal metabolic rate. Orangutans have a lower BMR compared to humans and chimps, as you see here in this figure from Ponser and colleagues. And apparently they have the lowest basal metabolic rate of any mammal that's been measured thus far, except sloths. Now, despite having all of these adaptations to low fruit conditions, there's evidence that Bornean orangutans are still adversely impacted by fruit scarcity. Low fruit periods have been associated with declining ovarian hormone production, lower conception rates, negative energy balance, ketosis, or the catabolism of body fat stores for energy, and negative protein balance. Previous research from Vogel and colleagues in 2012 indicated that prolonged periods of low fruit availability are associated with increased levels of delta N15 in orangutan urine. That's right there on this graph, indicating that muscle tissue wasting may have started to occur. Orangutans are argued to have an enhanced capacity for fat storage, which is evidenced by their propensity toward obesity in captivity. So this is a pretty remarkable possibility that fat reserves may be utilized to a point that the body resorts to catabolizing functional body tissues instead. So our aim was to evaluate this possibility by assessing variation in lean body mass relative to fluctuations in fruit availability. Because orangutans experience reductions in caloric intake when fruit is scarce, we predicted that episodes of fruit scarcity would be associated with skeletal muscle wasting. In other words, they would lose muscle mass when fruit availability was lower. Based on general differences in body weight and size, we also expected muscle mass to vary among the different age sex classes of orangutans, with flanged males having the highest muscle mass, followed by unflanged males, adult females, then subadult individuals. This is fairly logical, of course, just based on observing differences in body size, but it's also the pattern that was found by Emery, Thompson, and colleagues for wild chimpanzees. Data were collected at Tuanan Research Station in Central Kalimantan from 2009 until 2017. A fruit abundance index is determined each month as the percentage of stems that are monitored that are currently fruiting. Urine is collected from first morning voids and specific gravity was measured using a handheld refractometer right there upon sample collection in the field. Samples were then frozen and analyzed later for creatinine in Erin Vogel's lab at Rutgers. A total of 1,130 samples were used in our analysis after we excluded overly dilute samples, and this came from 70 different individuals. To estimate lean body mass or muscle mass, we used a method that's been validated in wild chimpanzees by Emery Thompson and colleagues, and has subsequently been used to evaluate muscle mass in wild capuchin monkeys by Bergstrom. So creatinine in urine and specific gravity are both measures that are used to control for the concentration of a urine sample, but creatinine is dependent on the muscle mass of the individual while specific gravity is not. So the variation in creatinine that goes unexplained by the variation in specific gravity then represents an estimate of muscle mass. So we then took the creatinine residuals and used them as a target in a generalized additive mixed model with fruit availability and age sex class as fixed effects and individual ID as a random factor. Estimated lean body mass was significantly predicted by both age sex class and fruit availability. You can see here that as fruit availability increases, so does estimated lean body mass for all different age sex classes. And while estimated lean body mass varied among the age sex classes in the general predicted pattern, with significantly greater muscle mass for males compared to all the other age sex classes, some surprising findings did emerge, which I'd like to talk about. This table shows the results of our GAMs using the releveling function to compare all of the different age sex classes to one another. You can see that adult female muscle mass was not significantly different from that of adolescents or dependent offspring. And while it's not terribly surprising that adolescents and adult females are not that different from each other, it would be surprising if adults didn't have more muscle mass than dependent individuals. So this is likely due to the small number of samples that we had for dependent orangutans. It's really hard to sample those little guys. And while adult males had higher ELBM compared to others, flanged and unflanged males were not significantly different from one another, contrary to our prediction. So as I mentioned already, that episodes of fruit scarcity were associated with a reduction in estimated lean body mass. What surprised us is that the patterns were consistent across all age sex classes. We reran the same model as a GLMM instead, with fruit as a binary predictor with high and low fruit periods so that we could visualize this. And all of the different age sex classes showed this decrease in muscle mass during low fruit. So what do these findings mean? These results show that despite several well-documented adaptations that orangutans have to cope with unpredictable fruit availability in their environment, they still enter a critical state of muscle catabolism when fruit is scarce, and all age sex classes experience this muscle loss. This raises questions about the potential implications for development. Orangutans have incredibly slow life histories, and it would be interesting to know whether individuals that experience longer or more profound muscle loss during infancy or the juvenile period see different developmental trajectories, for instance. It's also worth noting that Tuanan is a non-masting forest, so orangutans here experience overall lower fruit availability, but more consistent patterns of fruit availability compared to sites like Gunung Palung that experience periodic episodes of incredible abundance interspersed with really severe fruit shortage. It would be interesting to compare patterns of muscle loss between different study sites to better understand the variation in orangutan physiology in relationship to their local ecologies. And finally, these findings really emphasize the challenges that this critically endangered primate faces. As global climate change threatens to amplify El Niño events, droughts and wildfires may worsen, having detrimental impacts on orangutan habitat and their food supply. Monitoring muscle mass over the long term may be another way to assess health status in conjunction with various ecological threats and changes. And this is a fairly accessible method for people to use to monitor populations.